People are different in almost every aspect of their being. The way they look, talk, walk, and even down to the expression of their genes. Almost everything is different. This is also true when we talk about diseases. Our susceptibility towards a disease and our response against it vary from individual to individual. This is attributed to the uniqueness of one's genomic and proteomic constitution. This led to the rise of personalized medicine, which aims to tailor disease treatments based on one's genome structure, lifestyle, and environmental conditions. Several tools have been developed to improve the field of personalized medicine, one of which is the use of induced pluripotent stem cells, commonly known as iPSCs. But before we move on, let's backtrack and explore the basics of stem cells. Stem cells are undifferentiated cells with the potential to become specialized cells. They originate from two main sources, the embryos and adult body tissues. Embryonic stem cells are classified as pluripotent. They are capable of differentiating into all derivatives of the three primary germ layers, ectoderm, mesoderm, and endoderm. Meanwhile, adult stem cells are classified as multipotent. They can only differentiate into specific cell types, depending on the type of tissue where they reside. I, together with my team Joanna, Razal, and Josh, will discuss iPSCs, its production, applications, advantages and disadvantages, and its future directions.
iPSCs are adult cells transformed to behave like embryonic stem cells. Similar to the pluripotent cells, they can become any cell type in the fully formed body except the placenta and umbilical cord. However, these cells are not obtained from blastocysts. Instead, they are engineered in the laboratory through the conversion of tissue-specific cells like your skin cells, liver cells, and blood cells. Probably you're now thinking, is this possible? How can a differentiated cell become a pluripotent cell again? Can iPSCs be used to generate new liver cells or new skin cells from differentiated blood cells? And my answer to all those questions are yes.
And now tackling about its production, in 2006 Yamanaka and Takahashi made what you previously thought impossible possible. Going back to the early 2000s, these two Japanese scientists took the stem cell community by storm with their experiment on adult fibroblasts from mice, which led to the discovery of iPSCs. So how did they do it? The answer is in the expression of selected transcription factors. Initially, they selected 24 candidate genes as possible factors that could induce somatic cells to become pluripotent. Out of these 24 genes, 4 genes such as OCT3 and 4, KLF4, SOX2, and CMYC, also known as OSKM, emerged to be responsible for producing embryonic stem-like cells in mice. And concurrently in 2007, James Thompson from the University of Wisconsin found that another set of 4 genes, OCT4, SOX2, NanoG, and Lin28, are sufficient to reprogram human somatic cells into pluripotent cells. These transcription factors regulate cell cycle and developmental regulation pathways, which are important for the critical induction of pluripotency. And because these cells were produced from genetic manipulation of differentiated cells, they were eventually termed as induced pluripotent cells. And to date, iPSCs are not only produced from fibroblasts, but also from hepatocytes, keratinocytes, astrocytes, mesenchymal cells, pancreatic cells, and peripheral blood cells. But the story of how iPSCs were produced doesn't only include the works of Yamanaka and Takahashi. The development of iPSCs were born from the compounding efforts of several researches that paved the way for iPSC development. From these researches, we highlight 3 important studies that greatly contributed to iPSCs. In 1962, reprogramming by nuclear transfer was discovered by John Gurdon as a possible form of somatic cloning. Using this technology, he found that the memory of nucleus can be erased. He proved this by injecting nucleus of adult intestinal cells from frogs to an inoculated oocyte, which gave rise to a cloned tadpole. This implies that differentiated cells contain the genetic information required for organismal development. And fast forward to 25 years, another scientific breakthrough was reported by Harold Weintraub, who discovered that cell fate was directed by master transcription factors. He observed that overexpression of MyoD, a mammalian transcription factor, can convert a mouse fibroblast into a myofibroblast. This phenomenon was further termed as transdifferentiation, which emphasized the presence of a master regulator, a transcription factor that determines and induces cell fate. And lastly, appropriate culture conditions in maintaining embryonic stem cells was optimized in the late 1980s by Austin Smith. He and his colleagues reported that leukemia inhibitory factor and basic fibroblast growth factor are necessary for the long-term maintenance of mouse and human ESCs respectively. All these three elements contributed to the successful development of iPSCs.
And now fast track to 13 years after its development, iPSCs have become well known in several fields of science. It has been transformed into a technology that efficiently reprograms accessible cell sources such as the skin and blood. It has also been developed to become a suitable technique for clinical use. This is why there has been a growing number of researches and applications that sought its use in the field of applied research and therapeutics. In fact, iPSCs have greatly contributed to the fields of disease modeling, regenerative medicine, and drug discovery. Do you know that years ago, human drug screening and disease models relied on patient samples and tumor-derived cell lines? Both of them have the potential of properly mimicking our body's response. However, patient samples have limited capacity to expand, whereas tumor cell lines contain genetic and metabolic abnormalities. These drawbacks restrict the use of these two models. But with the advent of iPSCs, disease-specific cells have been developed. And through the years, the number of diseases successfully modeled via iPSCs have increased constantly. In fact, we can already generate hematopoietic cell from fibroblasts and use it in studying thalassemia. We have also developed dopaminergic neurons from fibroblasts, contributing to Parkinson's disease studies. But the application of iPSCs do not end there. They have been used in regenerative medicine, wherein immune rejection is one of the biggest problems. This technique has also been applied in gene targeting, making it possible to repair cells affected by mutations. And lastly, iPSCs have generated models that can be used to evaluate the efficacy of novel drugs and to limit the use of animal models. And with these great applications in the clinical world, scientists are now looking into the anti-aging properties of iPSCs for other medical purposes. Who knows? No one's stopping you from getting that fresh look.
On the other side of the picture, iPSCs are also applied in the basic sciences. Earlier we mentioned the 4 core transcription factors responsible for producing pluripotent cells. These transcription factors are important in cell cycle and gene regulation, creating a link among cell reprogramming, developmental biology, cancer studies, aging studies, and animal biotechnology. Through the selective expression of transcription factors, normal and cancer cell lines have been developed for developmental biology in cancer researches. In vitro aging studies, iPSCs serve as rejuvenated cells, allowing the elucidation of the aging process, specifically the accumulation of genetic abnormalities and the occurrence of epigenetic modifications. And lastly, iPSCs in animal biotechnology allow genetic manipulation of animal cells for the production of certain enzymes and the validation of transplantation therapies through animal disease models. To date, the manipulation of iPSCs are also being explored for preserving endangered animals.
The application of iPSCs have indeed grown and is now competing with the functions of traditional fetal, embryonic, and adult stem cells. The primary leverage of these stem cells is their means of production. They can be sourced from several tissues of a patient and be transplanted back to the patient, thus preventing immune rejection. iPSCs also lack ethical implications, which is prominent in the use of ESCs derived from a human embryo. And overall, the production of these cells are robust and minimally invasive. Therefore, it is eyed to unlimitedly produce iPSCs for various applications in disease modeling, regenerative medicine, and drug discovery. But it is important to note that these iPSCs also have their disadvantages and limitations. They are reprogrammed with low efficiency and slow kinetics, and they are susceptible to genomic alterations. The culture systems used in iPSCs may also contain animal-derived products, which may be toxic exogenous antigens, viruses, and zoonotic pathogens. Another potential threat to using iPSCs is their tumorigenic features, which can be attributed to genetic and epigenetic causes brought by the variation in their somatic cell origin, stress, and culture conditions. Overall, these imply that iPSCs still require imperative actions to circumvent the several challenges and consequences that come with its use.
Researches on iPSCs have been evolving ever since. From 2007, it allowed the germline transmission of mouse iPSCs, to 2019, where direct cell reprogramming using iPSCs made transformation of one somatic lineage to another possible. iPSCs through the years have proven its potential in several fields, especially in medicine. And in the coming years, we are hoping to conduct clinical trials using iPSCs-derived cells, manufacture 2D cell sheets, 3D tissues, or even entire organs from iPSCs, and completely understand the fundamental functionality of the genome to generate a high-quality and well-characterized iPSC lines. We still have a long way to go, but with more research on iPSCs, we can get past these insurmountable challenges. Because 100 years ago, iPSCs were once considered impossible, but now, we are steps closer to maximizing its full potential.